Mushroom can heal you, can feed you, can kill you. It's not like vegetable, it's not like animal, but it's somewhere in between. If you are walking, there's about 300 miles of fungi under every footstep that you take, and that's all over the world. Macrofungi can be divided into three major ecological groups. Macrofungi can be saprophytes, organic materials decomposer. Macrofungi can be mycorrhiza, generous fungi that give nutrients to their bias trees. Macrofungi can also be parasite, causing fatal and selfishly killing trees for the sake of food. Let's study further together about these three ecological groups of macrofungi with a study conducted in UNIVERSITI TEKNOLOGI MARA, campus JENGKA. The latitude of this university was 3 degrees 45 minutes 17 seconds north. The longitude was 102 degrees 33 minutes 43 seconds east. Hi, my name is Nur Amira Sakina binti Mat Shafi with metric number ID 2019406008. I'm going to present about functional diversity of macrofungi from University Technology Mara UiTM in campus JENGKA. Macrofungi is a group of organisms that live on various substrates and carry different functions accordingly. They divided into three ecological groups. First is saprophyte. Saprophyte is fungi that help in breaking down and decomposing organic matter like dead plants and animals. Mycorrhiza is fungi that have mutual benefits relationship with plant, as the plant provided fungi with sugars and carbon, while fungi provided plants with water and nutrients. The third one is parasite. Parasite is fungi that possess enzymes that could harm living plants or animals to obtain nutrients. The functional diversity of macrofungi in UiTM JENGKA has never been reported, although some macrofungal diversity was reported before. Specimen collection have never been done before in UiTM JENGKA. Currently, macrofungi are facing challenge, which is an extreme climate change. If this condition persists, the functional diversity of macrofungi that inhabit in UiTM JENGKA areas will remain unknown. This research provided data on the functional diversity of macrofungi, either saprotroph, mutualists, or parasites. Macrofungi in UiTM PAHANG main campus. There were likely novel or untapped species of macrofungi that live in the campus areas that could be recorded for future reference. This finding will be beneficial for a new documentation of macrofungal species in PAHANG as a whole. The objective of this study is to collect and to identify the macrofungi in University Technology Mara JENGKA PAHANG. The second objective is to determine the functional diversity of macrofungi from the university. There were 37 species of macrofungi identified with 21 different families which belong to the phylum Ascomycota and Basidiomycota. 31 out of 37 species founded were Basidiomycota. Basidiomycota, which was only consist by agaricomycetes, made up 84% of the total sample. Ascomycota made up 16% of the whole sample. It can be divided into two classes, namely Sordaromycetes, 11%, with 4 species of Pezizomycetes, 5%, with 2 species. The functional diversity of macrofungi recorded showed the prominence of decomposer groups and mycorrhizas. In the campus area, saprophytic macrofungi accounted for the majority of functional diversity, which were 27 species, which made up to 73%, then followed by mycorrhizae with 8 species, 22%, and lastly 2 species of parasitic, which made up to 5%. UiTM JENGKA has a lot of chopped trees and branches, hence providing the favorable environment for saprophytic fungi to grow. Saprotroph could degrade a variety of resistant organic substrate found in some land use systems, such as natural and planted tree. There were 4 species of agaricus found in the university area. Around the world, agaricus species are widely dispersed in both temperate and tropical regions. Species in this genus not only act as decomposers, but are also crucial as food and medical sources. The smaller number of mycorrhizal fungi found probably because the types of trees that were planted in UiTM JENGKA did not require interaction with macrofungi. A species of Physolithus arizus was discovered in this research. Physolithus species develops ectomycorrhizal connection with a variety of woody plants and was a global species that may be found in both tropical and temperate climates. Since many different species of trees have been involved in the development of Physolithus species basidiums, the host range of these fungi has been regarded as being relatively broad. Parasitic fungi also require only specific nutrients on specific trees to grow. Therefore, restricted ranges of hosts have restricted the diversity of this type of fungi. 2 species of Ganoderma species was discovered in this research. Ganoderma species, which are the main causal agent for basal stem rot on oil palm trees, have a hybrid lifestyle of biotroph and necrotroph. However, the hosts of Ganoderma sp in this research were not oil palm tree, but gelam tree and an unidentified ornamental tree that was planted by the roadside of UiTM JENGKA. UiTM JENGKA is a favored habitat for saprophytic macrofungi. Saprophytes were the highest collected macrofungi in this research, which was 73%. Several plants form mutual symbiotic relationship with fungi in UiTM JENGKA, as there were 22% of mycorrhizal fungi were found. Parasitic fungi in UiTM JENGKA was just 5%. This might be because there were not many trees suitable to be their host in UiTM JENGKA. All collected macrofungi were only determined by macromorphological characteristics like cap color and surface of the macrofungi. However, for a better species identification, micromorphological characteristics should be included. Species only recovered from a small number of sampling sites. Less than 10 sites were selected for the sampling sites. UiTM JENGKA is 1,000 acres and many other sites that can be explored to collect more specimens. The recommendation is use of molecular approach by using specific genes to assist the identification of the species, as most of the species were identified to the genus level only. Studies on chemical and medicinal properties of the macrofungi in the UiTM JENGKA could be done in the future to point out which fungi are useful and beneficial. The edibility of macrofungi also should be studied as additional information.
Thanks for watching.